So, like Sam said, I encourage everyone to type their questions in the chat. I'm not sure if I'll answer them as we go or at the end. It might depend on how complicated the question is, but please go ahead and do the questions as we go. So I'm going to be talking today about sort of regulatory issues surrounding adaptive design clinical trials. Does my voice still have echo? Are other people experiencing that as well?
Dr. Scott, you sound okay to me. I think you can continue.
Okay, I'll go ahead. All right, so I'm going to be talking about regulatory issues surrounding adaptive design clinical trial proposals. I did want to stress that this is just my own perspective. There's no one regulatory perspective on adaptive design, and if there were, it probably wouldn't be mine. But you know, I will sort of try to address how different parts of the FDA look at adaptive design proposals and what our guidelines are for evaluating them. I have nothing to disclose. I do have something to disclaim, which is that my presentation reflects my own views and shouldn't be interpreted as FDA policy. So I have three basic goals today. The first is to explain FDA's current thinking on confirmatory adaptive design. Second is to dispel a couple myths. I'm not sure if these are widely held myths, but I have heard them before. One, that FDA doesn't accept adaptive design trials, and two, that FDA doesn't accept Bayesian methods. And then finally, I'm going to try to give a little bit of advice on moving adaptive design proposals toward regulatory acceptance, on sort of advice on interacting with FDA. Okay, so the regulatory basics for adaptive designs. The first thing I wanted to stress is that FDA is not sort of one monolith in terms of our approach to adaptive design proposals. There are three FDA centers that are largely autonomous that are responsible for regulating medical products for human use. That's the Center for Devices and Radiological Health, regulates most medical devices. The Center for Drugs Evaluation and Research regulates most drugs and some biologics. And my center, the Center for Biologics Evaluation and Research, regulates the rest of biologics, as well as some devices and a handful of drugs. And because of this structure, it's entirely possible that the same adaptive design proposal, maybe even in the same indication, could get three different results from the three different centers. And there's several reasons for this. There are different laws that govern the three different centers. There are different regulations, different guidance documents, which I'll be getting to a little bit later, and sort of the least tangible, but in some ways maybe the most important, is maybe different cultures at the centers. Even within a center, there's not necessarily going to be one monolithic approach to adaptive design. Each of the centers is organized in various ways into review offices and divisions, usually by indication or by type of product they regulate. And each of those divisions has considerable autonomy and may have different attitudes that are governed by the indications they work with, the product classes they work with, or in some cases, concerns about a specific product. And fundamentally, the sort of bottom line is that every proposal gets reviewed by people. And people are individuals and have individual viewpoints. Having said that, I don't want to suggest that it's a free-for-all and you'll get arbitrary decisions from FDA. All of FDA's decisions are supposed to be backed by science and law and should be rational and somewhat predictable even. So in terms of the regulatory basics for drugs and biologics, that's for CDER and CBER, the major interactions with FDA occur in the context of either investigational applications or marketing applications. And the marketing applications that I'm going to focus on at least are new drug applications for drugs and biologics license applications for biologics. In terms of investigational applications, both drugs and biologics come under IND, which stands for Investigational New Drug Application. applications. I apologize for the alphabet soup sort of throughout all of this. It wouldn't be government if there weren't acronyms. Devices are a little bit different. The regulation of devices relies on classifications that are based on the risk of the device, or maybe the presumed risk of the device. So class 1 devices, which are considered, I'm not sure what the statutory language is, but basically harmless. These are usually marketable without pre-market notification. The classic example of a class 1 device is a tongue depressor. A class 2 device is something that requires pre-market notification, which is a kind of application to the FDA. It's usually called a 510K application. There are different forms for that application, but it usually takes the form of showing that the new device is substantially equivalent to a device that's already on the market. And then the most risky devices, class 3 devices, those require pre-market approval. So this would be a lot of implants, devices that carry a real risk of morbidity or mortality associated with their failure. So pre-market approval, those are PMA applications. For devices, the investigational applications are called IDEs, which stands for Investigational Device Exemption. So back to drugs and biologics. When someone submits an application for a new drug or a new biologic to be licensed, there's basically 3 possible outcomes. The first is that the product is approved or licensed, and then there's a lot of discussion about the form of the packaging and the labeling information that goes along with that. Another possibility is that the product is not approved, and FDA will send the applicant something called a complete response letter, which is supposed to be a roadmap to what needs to be done to get the product approved. And the third possibility is that the application isn't even filed by FDA. If FDA on initial review feels that the application isn't complete enough to review adequately, sometimes they won't review it. Now, which of those 3 outcomes someone gets obviously depends quite heavily on how FDA views and interprets the design and conduct, especially of the phase 3 or the confirmatory studies that support that application. And there's many, many years of precedent for accepting a really wide variety of different, let's say, non-adaptive study designs or traditional study designs. But if you want to propose, especially a novel adaptive design, it might need to break new ground in terms of licensure. This slide got a little bit cut off, but it's not important. In order to have a drug or biologic approved, it has to be shown to be safe and effective. And from the regulations, effectiveness is shown by evidence consisting of adequate and well-controlled investigations, which we sometimes abbreviate ANWC. The S on the end of investigation has been the source of a lot of almost Talmudic scholarship over the years. And it's usually interpreted to mean that you need at least 2 phase 3 trials to get a drug approved. There are a lot of exceptions to that. It depends on the indication, but that's often the default attitude. So because of this statute and because of the regulations, it might be important to think that FDA thinks your trial design is potentially adequate and well controlled. But, and this is a point I want to stress a lot, that's only if you have some design to bring a product to market. If your research doesn't have some ultimate commercialization in mind, it's not necessarily that important to you whether or not FDA thinks the trial is adequate and well controlled. So that's a kind of common misconception, and I did want to straighten that out. I also wanted to mention that there's a strong convention. It's not law or regulation, but a strong convention that to demonstrate effectiveness, type 1 error has to be controlled at a 2.5% rate. So INDs. So INDs are the applications you need to run a clinical trial with an investigational drug or biologic. And it's more likely you'll be interacting with FDA over INDs than BLAs or NDAs. So formally, you need an IND to transport or distribute an unapproved product across state lines. And usually that's done for research purposes, for clinical trials. There are other reasons. There are treatment INDs or compassionate use INDs, but mostly we're talking about research studies. And this doesn't include all research. So there are a lot of examples that don't fall under IND authority. A lot of comparative effectiveness research, for instance, involves comparing two or more approved agents in the indication that they're labeled for, and that generally doesn't require an IND. I would say, your mileage may vary. If you're not sure your study doesn't require an IND, then certainly you should contact the relevant part of FDA to find out for sure. Now, the FDA review of an IND protocol, in one way, it's a binary decision. There are two choices. Either FDA puts the protocol on clinical hold, which means you can't run the trial, or it doesn't put the protocol on clinical hold. But in either of those cases, there will often be a lot of comments and advice from FDA on either how to improve the trial, how to make the trial more likely to support eventual licensure of a product, or in the case of a clinical hold, we're required to send comments telling you what you need to do to get out of clinical hold. So according to the regulations, there are a lot of reasons a study can be put on clinical hold, but almost all the time it's one of two reasons. The first is that FDA feels the study puts subjects at an unreasonable and significant risk. That applies to all kinds of trials, all phases of investigation. And the second reason applies only to phase 2 and 3 trials, and that is that the trial is seen as clearly deficient in design to meet its stated objectives. So if FDA, let's say you submit an adaptive trial to FDA, and the reviewers are not confident in it, they're not sure that it is completely rigorous or that it controls type 1 error at exactly the rate it says it does, or some concern along those lines, FDA could, in theory, put that trial on clinical hold because of the clearly deficient in design standard. But what would be much more likely to happen is that FDA would allow the trial to proceed, but would provide comments noting reservations about the trial. And this gets back to what I was saying before about mostly caring about FDA's opinion when there's commercialization in mind. Those reservations will take the form of, we are not sure that because of issues X, Y, and Z, that this trial can support approval of a product. If you're a pharmaceutical company, that language is terrifying. If you're an academic investigator, that might or might not matter to you. I wanted to talk about special protocol assessment. This is a special regulatory pathway where applicants can request a formal written agreement with FDA about the design and size of a clinical trial that's intended to form the basis of an efficacy claim for a drug or a biologic. And I wanted to stress that this is not usually the case. So if an IND is submitted and it's not placed on clinical hold, that's often interpreted as being in some sense a blessing from FDA. And it is at least a very mild blessing in the sense that FDA doesn't think it needs to be on clinical hold, but it doesn't constitute a formal agreement with FDA that when your trial is over and you submit a BLA or an NDA, that FDA will approve the product. But an SPA is closer to that. That's not exactly what an SPA is either, but it is a formal agreement that the trial is adequate to provide evidence of effectiveness. There's a question, does SPA apply to devices as well? No, it doesn't, but there is a similar program which is called, okay, I don't remember what it's called. It's called something consultation, and it's very similar. Unfortunately, I know drugs and biologics much better than devices. Sorry for not being able to give a more precise answer to that. So obviously, if you have a novel adaptive design, it would be very comforting to have an SPA for that design, but it's not always the best choice to even seek it for two reasons. One is that the individual review divisions have a lot of discretion in terms of granting SPAs. And some rarely do it, some do it all the time, but it's not, it might be a tough sell if it's a very novel proposal. And the other issue is that FDA has very strict review timelines with SPAs. When an SPA is submitted, we have 45 days to concur or not to concur with it and to provide reasons. And a novel design might take longer than that to review. And what that will mean in practice is potentially many iterations of review. Whereas if it was just under IND, after 30 days, if you're not on hold, you can go ahead with your trial. Okay, so the next bit I'm going to talk about are the guidances from FDA on adaptive design. This did not used to be plural. I gave a very similar talk to this about 2 years ago, and at the time we only had one draft adaptive design guidance. Now we have two. So these are the front pages of those guidances. On the left is the adaptive design guidance for drugs and biologics. Draft, I should say, and it was released in February 2010. So it's been a draft guidance now for more than 5 years. On the right is the draft guidance for adaptive design for medical device clinical studies. And that was issued just a few weeks ago. It's hot off the presses. And in fact, it's still in the public comment period, which I'll talk about later. One thing I like to point out, the drugs and biologics adaptive design is signed by CDER and CBER, and the devices adaptive design guidance is signed by CDRH and CBER. So at CBER, we literally have two different guidances that potentially affect our behavior. Hopefully they don't disagree too much. So first, what is a guidance? FDA's actions are governed by sort of cascading levels of policy. At the top, I didn't put this in, but at the top is the Constitution. And then there are statutes that are enacted by Congress. And those are obviously binding laws subject to judicial interpretation. Then there's regulations, which are considered binding interpretations of law by the FDA. And then a level below that are guidance documents. Guidance documents are non-binding descriptions of FDA's current thinking about a topic. Typically, before we release a guidance, there will be a public workshop discussing the issue. And guidances are generally released as drafts for public comment. I think usually for 90 days. The devices guidance is you have 60 more days to make comments on it if you want. And those comments have to be addressed before the guidance becomes a final guidance document. And I mentioned the drugs and biologics adaptive design guidance has been a draft for 5 years. Not coincidentally, they received over 900 comments on that guidance. So I think there's a causal relationship there. So starting with the drugs and biologics adaptive design guidance, it focuses on confirmatory trials, almost exclusively on confirmatory trials, or adequate and well-controlled trials in the language of the regulations. And it defines adaptive design clinical studies as a study that includes a prospectively planned opportunity for modification of one or more specified aspects of the study design and hypotheses based on analysis of data from subjects in the study. And the thing I would stress in that sentence is prospectively planned. An adaptive design is something that involves planned modifications. It's sort of maybe surprising for some people to hear it, but we very often will have people come in wanting to just change stuff in midstream of a clinical trial, which happens, things come up that you didn't expect, and refer to that as an adaptive design. And we do not consider that an adaptive design unless it was planned from the beginning. Adaptive designs are documented in detailed protocols and usually also in separate statistical analysis plans. So some things that are not adaptive designs. First of all, modifications based on non-pre-specified analyses of interim data, or I should say either the interim data analyses weren't pre-specified or the modifications weren't pre-specified. And most of the time, when we get a proposal like this, which, as I said, is not uncommon, our default position is almost always going to be no, and then there might be reasons to change our mind about that. So sort of in a way, the burden of proof is on the applicant that those changes are necessary and won't permanently damage the conduct of the trial. Another class of things that aren't considered adaptive designs are modifications that are based entirely on external information to the study. So for instance, maybe you're running a study in glioblastoma and another very large clinical trial in glioblastoma is published, and it teaches you something about what your endpoint should be or what your inclusion-exclusion criteria should be, and you want to make a modification based on that. That's also not considered an adaptive design, and in many cases, that kind of change is fine. But there will be a requirement to show that there was no peeking at the data, and also everyone will have to be assured that it won't permanently, really seriously damage the conduct of the trial. In terms of exploratory studies, the guidance talks about the difference between adequate and well-controlled and exploratory studies. And while it doesn't say you can do whatever you want in an exploratory study, it does strongly encourage experimentation with adaptive and other novel designs in exploratory studies. And a couple examples of adaptive design methodologies that we see fairly frequently in earlier phase studies are CRM designs for dose finding in phase 1 and selection designs for dose selection in phase 2. The big concern with adaptive design in the exploratory setting to FDA is avoiding the trap of thinking you're more sure about something than you really ought to be as you go into phase 3. But other than that, there's a lot of leeway. In terms of what can be adapted, as far as I know, anything can be adapted in theory. I don't know anything that's ruled out in terms of adaptation. Some things that are listed as examples are eligibility criteria, randomization procedures, treatment regimens, sample size, follow-up schedules, endpoints, analytical methods, various things, and so on and so on. What the guidance says is when FDA is reviewing an adaptive design proposal for a drug or a biologic, there's basically 3 categories of general concerns. And the first is the one that we pay the most attention to, I think. I'm not sure it's the most important, but it's important. And that's the risk of false positives. Hopefully most of you are familiar with the idea of multiple testing leading to the inflated possibility of a false positive or a type 1 error. And the sort of very basic heuristic for looking at that is the more ways you have to win, the more ways you have to come to a positive decision, the more chances you have of making a false positive decision. And in some cases, it's very easy to control for that, and in some cases, it's extremely complicated to control for that. Another issue related to false positives is difficulty in interpreting results of adaptive design trials after they have been successful. So in some cases, even after we've controlled for multiple testing in terms of type 1 error, it's not always clear exactly how to do estimates of effect size for the adaptive design, or especially interval estimates, confidence intervals or credible intervals for the adaptive design to let us really get a scientific interpretation of its results. Another issue that comes up in certain kinds of designs are populations themselves can be moving targets. Sometimes eligibility criteria will be expanded or narrowed as the trial goes on. A subgroup might be dropped, for instance. And then at the end of the trial, there's going to be a lot of discussion and a lot of, in some cases, confusion about whom the results apply to, and if the drug were approved, how would you write the indicated population statement in the label? So that's another real issue. Finally, people are concerned about operational biases. A lot of adaptive designs require unblinded analysis of data for various purposes, and there's a general category of concerns that knowing things about the trial as the trial is ongoing can affect the conduct of the trial subsequently. So the a sort of simplistic example of that would be if an interim analysis suggests that you can reduce the sample size, then people will interpret that as meaning that the investigational agent is likely doing pretty well, is likely pretty effective, and that might affect the way people handle treatment assignment or who they bring into the trial, how they do screening, various things like that. And so there's a lot of importance attached to documenting who knows what about the trial and when do they know it. I'll talk a little more about that later. The next general category of concerns with adaptive designs discussed in the guidance are false negatives. So it's sort of conventional wisdom that adaptive designs are more efficient or more powerful than conventional or static designs. And that's not necessarily the case. So there can be a chance of inflating the risk of a false negative. One source of this risk is not exactly statistical, it's more scientific, which is that when you're using a very efficient design to sort of rush through development, and especially if you think of like a seamless phase 2-3 design, where you're designing the phase 3 trial before you've performed the phase 2 trial, that can, in some cases, really limit the changes you might make to the phase 3 trial after you've seen the phase 2 trial results, which can reduce the likelihood of success of phase 3. And the third category of general concern is time. Again, people usually think of adaptive designs as increasing efficiency, but it's also important to recognize that there can be an additional time burden in terms of planning and designing the trial, and also in terms of interactions with FDA in some cases, especially if it's a special protocol assessment. But even if it's not, novel proposals usually will get more scrutiny from FDA for understandable reasons. And so you might need to build in more time for more extensive communication between FDA and the investigator or sponsor. I think the guidance notes that as we get more experience with various kinds of adaptive designs, this particular concern should more or less melt away. But I think people will always think of new things to do, so there's always going to be some time issues. One of the more controversial parts of the draft guidance is the use of this term "well understood." The draft guidance divides trials into two basic categories. It calls trials either generally well understood adaptive designs or less well understood designs. And I don't care for this language very much. A lot of people don't like it very much. And just on the surface level, I don't think the guidance really should be making statements about, the cognitive status of the reviewers of the design. Some people might understand a design very well, and some might understand even a conventional design not so well. And what these terms mean in context, and I think the way I would have written it or would re-express it if I had the chance, is designs for which we have a lot of experience and designs for which we don't have a lot of experience. So generally well understood design would be a design for which FDA has substantial experience and is comfortable with its use in an adequate and well-controlled trial. And less well understood would translate to FDA has less experience, or we're not confident that error rate inflation, operational bias, or other issues are under control. But sticking with the language of the guidance, in terms of what a well understood design would be, the guidance talks about almost any adaptation that's blinded to treatment effect. So anything based on blinded analyses is generally well understood. And also, quote unquote, traditional group sequential designs, things like Land-Demet's spending functions, even SPRT designs, although we don't see a lot of those. Those are considered well understood. So some examples of well understood designs would be adapting eligibility criteria based on baseline data of the trial, re-estimating sample size based on blinded analysis or pooled analysis, adaptations that are based on outcomes that are unrelated to efficacy. That one has always confused me a little bit because I think usually outcomes are related to efficacy. Group sequential designs that are implemented by data monitoring committees are considered well understood, and adaptations that are based on other things that are unrelated to treatment effect, like the amount of missing data or the pooled data distributions, those are considered well understood. In contrast, less well understood designs are designs that are based on unblinded interim analyses of treatment effects, not counting traditional group sequential designs. So group sequential designs are based on unblinded interim analysis of treatment effect, but those are considered well understood. So there's a carve out for that. So examples of things that usually fall under this category are dose selection designs, response adaptive randomization, although not covariate adaptive randomization, unblinded sample size re-estimation, changing populations or endpoints based on treatment effects, any combination of techniques to get to sort of very complex designs. And also the guidance talks about adaptation in non-inferiority studies as being an issue of little experience to date. And remember, to date was 2010. I think we've probably seen some more of those by now. Some other considerations the guidance talks about are the implications for safety analyses, the content and format of documentation for adaptive designs, interactions with FDA, simulations, having SOPs in place for data integrity, blinding, and information sharing, and reporting the results of adaptive designs. So that was the drugs and biologics guidance. I'm going to talk a little bit about the devices guidance. As I mentioned, this is a new guidance as of May, and it's very similar, I think, in spirit to the drug and biologic guidance. I don't think it contradicts the drug and biologic guidance, at least not in any major point. The main differences in the device guidance is that it, first of all, builds on an earlier guidance for the use of Bayesian statistics in device trials, and so it has a lot more explicit discussion of Bayesian methods for adaptive design. It doesn't use the terminology less well understood, and it uses device terminology instead of drug and biologic terminology. I think, just to take a simple example of that, I believe the drug and biologic guidance, the title has the word trial in it, and the device guidance has the word study in it. And finally, the device guidance talks about some things that are of either unique or particular importance in device trials. So those topics include what they call adaptations of the device itself. I don't love this language either. I would call it modifications of the device, which is not really that uncommon in device trials. You find you need a slightly bigger or smaller catheter. You have to make little changes to the device as the trial goes on. That's discussed in this guidance. It also has sections discussing open label adaptive trials, observational adaptive studies, adaptive design in single arm studies, and adaptive design for diagnostic studies. I mentioned on the previous slide that there was an earlier guidance on Bayesian statistics for device trials. It covers a lot of related topics and is a good sort of cross-reference. Okay, so that's it for that section. Next, I'm going to be talking just sort of briefly about some of our experiences at the Center for Biologics with adaptive design submissions. So we performed a survey of our IND and IDE reviews from 2008 to the end of 2013. We focused only on phase 2 to 4. In other words, we didn't look at phase 1s. During that time, there were about 12,000 submissions that were assigned to statisticians for review. And we ended up having 1,200 statistical review memos to screen. And of those 1,225, 140 involved adaptive design components, so a little more than 10%. We broke the results down by the product office within CBER, which for us means the office that deals with vaccines, the office that deals with blood products, and the office that deals with cellular tissue and gene therapies. So I mentioned earlier that I gave a version of this talk a couple years ago. When I gave a version of this talk a couple years ago, this graph stopped at 2011, and I said adaptive designs just keep going up. But since then, they have not gone up. 2012 and 2013, especially for phase 3 trials, there's been a dip in the number of adaptive designs. And I've seen similar results from an industry survey. I haven't seen updated results from CDER or CDRH recently, but they may have a similar experience. I don't actually have an explanation for this. I think maybe there was an initial flurry of interest in adaptive design after the first draft guidance was published, and maybe that's died down a little bit. But there's still a steady interest in adaptive design proposals. This slide summarizes what kinds of adaptive designs we've seen. The biggest bar here by far is group sequential designs. The next biggest bar is sample size re-estimation, that's SSR. And then there's also this sort of group sequential plus sample size re-estimation bar. Everything else, there's sort of bits of here and there. I do want to distinguish, there's a line here for Simon's two-stage. That's for two-stage designs in phase 2, usually in oncology. And then the other two-stage things below that, that's a different kind of two-stage. That's for usually phase 2/3 studies, designs that have both a learning and a confirmatory stage. This is a summary of the kind of endpoints. We tend to see response rates and time to event endpoints. It's not critical. And most of this table is also not critical. It gives sort of trial characteristics. A couple things I want to focus on. First of all, about 80% of the phase 3 proposals were frequentist designs. Only 2 proposals were pure Bayesian designs, and the other 15 were usually some hybrid, maybe a design that has Bayesian monitoring, but a frequentist hypothesis test and decision rule at the end. Most of the phase 2 designs that we received fell into the category of a well understood according to the guidance. In phase 3, only about half were clearly in that well understood range, and most of those would be the group sequential designs. And then finally, the review outcome. Sorry, there's a question. That one looks a little complicated. I'll try to get to that question at the end. I'm also not sure if I can answer it. So the final couple rows of this are the outcome of the FDA's review. In phase 2, about 50% of the time the trial was allowed to proceed without any further comments or requests for clarification. In phase 3, that was much less common. 3/4 of the time, FDA requested more details, usually, or requested changes to the study. A few notes about our adaptive design experiences. Most of our adaptive design proposals are in the cellular tissue and gene therapy area, especially in oncology. As I mentioned, very few are Bayesian. And after group sequential designs, the most common proposals are sample size re-estimations. So I don't have a lot of examples of approved products that were approved based on innovative adaptive designs. Most of those proposals are still under IND and have not come to fruition. The devices group, CDRH, has many more examples of approved products. And also, we see more experimentation in early phase than in confirmatory trial, which is sort of as expected and encouraged. I also wanted to talk a bit about simulation. I believe there was a previous webinar about the importance of simulation in this kind of design. The Bayesian device guidance talks about simulation at some length, and the Center for Devices considers simulated type 1 error rates to be fairly non-controversial. It doesn't mean they accept it all the time, but they don't consider the simulation in itself to be a problem. But the drugs and biologics adaptive guidance is a lot more ambivalent. It has this quote: using simulations to demonstrate control of the type 1 error rate is controversial and not fully understood. Um, and I will agree that it's controversial. This is something that we have internal discussions about a lot. And there are people who feel fairly strongly on both sides of the issue about whether simulations for type 1 error are a good idea or not. What I will say is that at the Center for Biologics, we have examples where we've accepted simulations for type 1 error, and each one is evaluated on a case-by-case basis. Some issues with simulations in this area is that these problems, if it's an interesting enough problem to have an adaptive design for, then typically the type 1 error simulations will be multidimensional and complicated, and it's not always clear what parts of the parameter space need to be explored. Even for the null hypothesis, there can be many different potentially interesting parts of the null space. Another issue is just in terms of FDA resources and expertise. Not every FDA reviewer has experience with these kinds of simulations, and because we don't live in a world where everyone who does simulations is using the same software package, it can be sometimes complicated and difficult for FDA to keep up. And finally, simulation error, stochastic error is an ongoing concern. And we usually like to look at confidence intervals for simulated type 1 errors. Okay, and the last part of this talk is sort of free advice on interacting with FDA. And the first bullet point here is really the main thing, which is to try to make FDA an ally in your development program or in your clinical trial. And what that really boils down to is if the FDA reviewers believe you're doing important, valuable work and believe that you're going about it in a good way, they will tend to do what they can to make that process go as smoothly as possible. And I don't want to suggest that we're stonewalling people that we don't agree with. That's not the case at all. But it's much easier to persuade someone to accept a novel design if they understand the rationale and the importance of that design in the context you're studying. And some sort of dimensions of that include very clear and adequate communication in your formal submissions and taking advantage of formal meeting opportunities with FDA, as well as informal contacts when possible. And in some cases, when you really can't come to an agreement with FDA, sometimes escalating the issue can be helpful. But also it can be time consuming. It's not always a good idea. Okay, so documenting a novel adaptive design proposal, obviously you need to describe the technical aspects of the adaptation very clearly, and that includes explaining both to statisticians and to non-statisticians. And successful proposals often have really two different levels of explanation to go along with that. Typically, a high-level explanation in the protocol and a very granular explanation in the statistical analysis plan. Pre-specification is always important to FDA for confirmatory trials. So, the first set of red flags that go up when we review a study are things that just haven't been adequately pre-specified. I've seen proposals where the extent of the adaptive discussion literally is, we plan on performing one interim analysis, at which point the sample size may be increased. And that's all it says. And that is clearly not enough pre-specification to control type 1 error. And speaking of type 1 error, FDA is definitely going to be looking for type 1 error control, so that should be addressed in the proposal. Another issue worth mentioning is describing the role of the trial in the overall development plan. And this comes out of experiences I've had where there's been a lot of back and forth about you know details in the statistical analysis plan between FDA and a sponsor, and then at some point the sponsor will mention, we see this as a phase 2 exploratory study, and if FDA had known that to begin with, there might have been much less discussion of those details. Also, documenting the chain of information passing for unblinded analyses is very important. So I already mentioned documenting the rationale. You don't have to document a rationale. There's no requirement for that. I just think it's a good idea in terms of sort of getting everybody aligned and on the same page about a proposal. I also talked a little bit about simulation already. The main lesson here is just to document the simulation adequately and to perform adequate simulations. And FDA will almost always ask for the simulation code, so you might as well submit it to begin with. And ideally, we would like to be able to run the code and run other scenarios using the code. And so there can be considerations about what kind of software or programming language environment you use. I mentioned formal meetings with FDA. For confirmatory adaptive designs, the really critical meeting is the end of phase 2 before phase 3 meeting. And the more prepared the applicant is in terms of having a good draft protocol, the more constructive those meetings tend to be. And even at the pre-IND stage, which in many cases means before phase 1, it can be useful to talk about long-term plans for a development program. There's also something called a Type A meeting available for stalled development programs, which basically means that sort of irreconcilable differences between the sponsor and the FDA. We can have a very high-level meeting to try to hash those things out. Informal meetings are also often possible. This really depends on the review division you're working with. Some do not grant informal meetings. Some grant them fairly regularly. And in all cases, it's more likely to be granted again if the review team feels that the research is important and well conceived. And you can also, don't harass. You can discuss scientific and adaptive design issues with FDA staff at public meetings. You shouldn't get a response about a specific submission, but you'll often find people are very happy to talk in general terms. And that's it. I wanted to thank several people from my division. Annie Lin is the person who actually did that survey that I presented, and Bozen and Estelle Russick-Cohen gave me some nice comments as well. And that's it. And we have about 10 minutes left, so I'm going to try to tackle this question. Could you talk a little about diagnostic strategy trials that involve a diagnostic device paired with an algorithm or a pathway? If the device is approved, but the pathway or use isn't. Yeah, so that is a good question, and I'm not an expert on this, but I can give you a little guidance. So some of the things you're describing would fall under the category of what we call companion diagnostic tests. So that would be in particular if there's a drug or biologic or a therapeutic device whose use is being governed by the use of the diagnostic. And the way that works in the regulatory framework is the Center for Devices will review the diagnostic, especially if it hasn't been cleared yet. But even if it has been cleared, they may need to review the diagnostic in the context of this specific use. And then the relevant other part of the FDA will review the drug or the biologic. Or if it's a therapeutic device, the Center for Devices will review that as well. We have a guidance on the use of companion diagnostics, which I recommend looking at for better information about this question. Another category of thing that's come up for us a little bit recently are the use of simply computer algorithms without a specific diagnostic. Well, I'm sorry, there is a diagnostic because there's an assay, but the use of software to tailor a therapeutic regimen for an approved product, that's come up with us recently. And that again is performed typically as a joint review between the Center for Devices and the center that regulates whatever the therapeutic modality is. If it's an unregulated therapeutic modality, then probably you just need to work with the Center for Devices. Are there any other questions? Or any follow-up on that one? So if a trial is a purely diagnostic trial, so diagnostics mostly are regulated by the Center for Devices. There are some diagnostics that are regulated here at CBER. Those include retrovirals and anything that's going to be used to screen blood donors. But most diagnostics are reviewed at CDRH. And in many cases, diagnostic trials do not require an IDE, an investigational device exemption. It depends, first of all, on the risk class of the device. So a class 3 device does require an IDE. A class 2 diagnostic, which most diagnostics are, usually the study doesn't require an IDE. But for specific advice, you would definitely have to contact the review division at CDRH or CBER. Okay, anything else? I see Sam. All right, well, thanks everybody for attending. I really enjoyed this.